Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
You probably never thought you had much in common with microbes such as Salmonella, Staph, or E. coli. But the truth is, you do. All living things, birds, fish, worms, lizards, grass, you, me, and all the microbes thriving underwater, underground, in the air, and inside our bodies, we're pretty much the same at the molecular and genetic levels.
Well, they're putting up a new building here. Not exactly sure what it's going to look like. Sure hope the architect knows. But what I can tell you is the fundamental concepts are the same on all construction sites. Raw materials are put together by builders to create new facades and infrastructures. And it's the same with living cells.
Whether they're algae, bacteria, fungi, plants, or animals, their raw materials, such as proteins and nucleic acids, are similar, and they combine together in ways which realize a host of possibilities.
I'm Jim Leinfelder. In this episode of Unseen Life on Earth, we'll explore not only the vast differences between living cells, but the cellular functions and structures that all living things have in common.
Those cells can look amazingly different. Each one faces the same challenge of getting energy and reproducing. Every cell needs nutrients and an environment specifically suited for its metabolic needs in order to carry out these functions and survive.
While a cell's internal architecture turns out to be different depending on whether it's a prokaryote or a eukaryote, all cell processes and products are similar and often identical.
Eukaryotes, like the cells of plants and animals, are designed like this office building with many floors and rooms. Eukaryotes are complex and compartmentalized, including structures such as lysosomes, mitochondria, ribosomes, and chloroplasts. They have an internal membrane structure and a true nucleus to enclose their DNA. Cell structures may differ slightly, but plants, fungi, and animals, including you and me, share the eukaryotic cell plan. Bacteria are prokaryotic. Prokaryotes are designed kind of like this baseball stadium. The floor plan has very few internal structures. Most prokaryotes have a cell wall. Some have a slime layer around the outside, but none have a distinct membrane-enclosed nucleus. The DNA is found free-floating, along with ribosomes responsible for protein synthesis. Several of the functions found in the membrane-bound organelles of eukaryotes, like mitochondria and chloroplasts, are also found in the cell membranes of prokaryotes. Many bacteria can move around using appendages called flagella. Membranes allow food in and waste out. They have all the necessary material and machinery needed to grow and reproduce. Proteins embedded in the cell membrane initiate duplication, following the code provided by the DNA strands inside. The cell then divides into two complete bacteria.
But even though prokaryotic cells can be as different in form and function from eukaryotic cells as an office building is from a baseball stadium, let's not forget, they do share common traits. They both have cell membranes and ribosomes and contain DNA as their genetic material.
In fact, the mitochondria and chloroplasts present in most eukaryotic cells are similar in dimension to prokaryotic cells. Coincidence? Not really. At some point in evolution, free-living prokaryotes took up residence inside primitive eukaryotic cells, becoming the first internal structures, or organelles. This process is called endosymbiosis. An abundance of microorganisms are hosted by the human body in areas such as the upper respiratory system, on the skin, and in the digestive system. The large intestine has hundreds of species of microbes living in communities. They exist cooperatively, digesting food and making vitamins.
Hi, thanks. Okay, got your eukaryotes like us. You've got prokaryotes like bacteria. They're all cells, they've all got membranes, DNA, RNA, but just when you think you've got it all figured out, nature throws you a curveball.
The common cold, influenza, chickenpox, and measles. You've heard of them. In fact, you might be carrying around the organism that causes one of these illnesses right now. But unless it's making you sick, you might not even know it's there.
Viruses. These tiny, I mean tiny, well, let me give you an example. Let's say the virus is the size of this baseball. Well, then a prokaryotic bacterium would have to be something considerably larger. Maybe the size of the pitcher's mound. And a eukaryotic animal cell would be about the size of the entire infield. Oh, right. Anyway, these tiniest of troublemakers may seem like they function like other organisms. But the truth is, they're not even considered to be cells. In their basic form, they're as inanimate as, well, this hot dog.
It takes contact between a living cell and a virus to bring it to life.
In fact, a virus is like a baseball in other ways.
Here you go.
Thanks. Hey, nice job. If you examine a baseball by, say, oh, I don't know, cutting it in half, you begin to get a sense of how some viruses are organized. The outermost layer of the virus, in this example, the horsehide on the baseball, is called the envelope, which is complex and can vary chemically. Some viruses don't have an envelope.
This next layer of tightly woven material here, that represents the capsid or protein coat, something which all viruses do have. And then this innermost rubber core, that represents the part of the virus that contains its genetic material. I'll take it from here, Bob, thanks. A virus can't replicate on its own. So because it lacks its own manufacturing plant, it has to invade a cell and use the host cell's materials and equipment to do the manufacturing. But what does this have to do with pizza, you're asking? Let's say that this pizza is a virus and it wants to replicate itself. It would have to invade this pizza parlor, which has all the ingredients and equipment, including me, that it needs to get the task done, just as a host cell has everything a virus needs to reproduce. A virus is the smallest and simplest of microbes. Unlike a bacterium, it doesn't have the ability to reproduce on its own. It must enter a living cell. Once inside, the virus sheds its outer protein coat. It hijacks the host cell's machinery in order to reproduce. The long chains of viral genes are duplicated by viral proteins made in the host cell. Once all the parts for the new viruses are completed, they reassemble. Within hours, a single infected cell can give rise to 1 million new viruses. They destroy the host cell and go on to infect new cells. The virus and cell interaction can sometimes cause disease. Humans aren't the only ones who can carry these tiny microbes. It turns out that all living cells can be invaded by viruses, from plants to pets and even bacteria. Viruses may seem like they don't have much in common with prokaryotes and eukaryotes, but actually, their chemical needs are quite similar. Although viruses all do the job differently, by borrowing a host cell's machinery, they all successfully achieve their goal of reproduction. And if this pizza were an actual virus, this parlor would be floor to ceiling in pepperoni and cheese.
All forms of life, prokaryotic, eukaryotic, viral, even construction workers, may seem different, and in many ways they are.
But most of their central processes are the same. They must all use energy as ATP, along with building materials to reproduce.
Enjoy it, gang. Baked it myself.
Oh, this looks good. All right.
Hey, thanks, man. They also have to store genetic information. Without blueprints, these construction workers wouldn't know what to build. The same goes for cells, only the blueprint, or genetic information, is in the form of DNA, or in some viruses, RNA. With a different blueprint, you get a different building, and with different nucleic acids, you get different organisms. The metabolic functions that all organisms have in common are the real-world expressions of their DNA. They're like the construction workers who take the building materials and put them together according to the blueprint to make the structure we see.
Scientists have discovered that all living things share parts of the same blueprint, shown by the common features present in all organisms. What we understand about our own species comes not only from studying our DNA, but also from studying the DNA of the microbes that evolved before us. Human beings haven't been around long enough to invent hardly anything new. We are largely built from the inventions of other species, including microbes. They came up with the inventions like shutting down for a few hours every day, which we inherited as sleep. as well as the basic mechanism that makes it possible for us to see. But even then, so what? One way or another, we have become marvelously capable, highly successful beings. And why on earth should you care about how it happened? To find out why you should care, take a trip with one of the people who is helping to decode your DNA. Meet Dr. Craig Venter. A consummate sailor, Venter and his crew are on their most ambitious journey ever. A voyage along the path that Columbus sailed from Spain to the Caribbean and the New World. Venter has been responsible for key breakthroughs in today's genetic revolution. After establishing whole genome sequences for many bacteria, his latest project is the sequencing of the entire human genome. Venter attributes his accomplishments to his different way of looking at the world.
I think each of the breakthroughs that I've been lucky to come up with my team have been because I go outside the box. It's much easier to go outside the box than trying to work up linear through a maze. You know, you go outside and you go around the traffic instead of trying to go through it all. And I think just by doing that, I've been able to get places and see relationships that other people don't see.
Some of the genes that make up the human genome have already been decoded by Venter and others. And perhaps the most remarkable things that research reveals is how much we can learn about the inner workings of your body from studying the inner workings of microbes.
It's clear every gene in the human body can be traced to genes in other species. We have 60 to 80,000 human genes. A bacteria has 2,000 genes, but we can trace back origins of all of our genes to things that we find in the archaea and in the bacteria.
Knowing the entire gene sequence of a microbe gives us the ability to compare our genes to other organisms. This has given rise to new insights into what happens when things go wrong inside us. Take colon cancer, for instance. It turns out that many people who get colon cancer are missing a gene. How did we discover that?
Those genes were discovered in terms of their function by aligning the sequences in the computer that we have obtained from human cells to those from the bacteria that lives in our guts.
Scientists found a gene in these bacteria which codes for an enzyme that repairs damaged DNA, a precursor to mutation. Then, Venter and a colleague compared the bacterial genome with a part of the human genome. They discovered the exact same gene in human beings and found that it helps keep us from getting cancer, also by repairing damaged DNA. People who are missing this gene can now learn that they are at risk long before cancer develops. Knowledge that will save tens of thousands of lives. What microbes are ultimately telling us is that each of us contains within our genetic material a written history of life on Earth. Take, for example, these human cells. From studying the DNA inside them, we have learned that cells like ours were born over 1 billion years ago by a marriage between different single-celled microbes.
Our basic cells are a combination of multiple organisms. We're composites of bacteria, lineages from bacteria and other microorganisms. So it's compounding life on top of life. Each genome is the recorded history of life.
After 15 days at sea, Venter and his crew arrive at their ultimate destination, the island of Antigua. In June of the year 2001, Venter expects to arrive at another destination, the completion of the sequence of the human genome. We stand on the verge of an unequaled explosion of knowledge about who we are and how we came to be here.
The ultimate point of most science is to try and understand who we are, where we came from, how we fit into the environment, how we fit into the overall picture. Did we extend out from life of these simple bacteria? Did we come from outer space? You know, who are we? What are we? You go out of this context of the laboratory and you go out and look at a tree or a fish or, you know, things I encounter on the beach, and I look at them and I realize that we share, you know, 90% or more of our genes with those other species.
Our new understanding of DNA has shown us that, beyond any doubt, we are beings whose roots lie in the 4 billion years of life on this planet.
Over 4 billion years ago, we didn't have intact cells. Life was a series of chemical reactions on the surfaces of metals, other things in the ocean, to eventually develop a cell that became self-replicating with the duplication of the DNA molecule. That eventually, over billions of years, became us, became all the other living species that are present on the Earth today.
But knowing this sometimes makes us uneasy, because ever since the first time we told a story about our origins, most of our stories have portrayed us as beings apart.
I find it empowering. I don't find it a threat to my humanity. I think it helps define humanity. With genetics, it's the intimate, minor differences that are everything.
In the coming years, we are certain to learn far more about the ways in which we're linked to the rest of life on Earth, and the ways in which we're unique. But no matter what lies ahead, it is unlikely that we human beings will ever again be able to view ourselves as a species apart. From one point of view, that may seem to diminish us. But from another point of view, we are gaining a deeper understanding of our role in what may be the most miraculous story in the universe. Life on Earth. And that new understanding can only serve to increase our sense of wonder. From the largest organisms to those that are rooted in the ground, to those that fly, swim, float, dig, to those too small for the human eye to see. All living things have common metabolic and reproductive processes. They store information and energy in the identical types of chemicals. We are all connected.
In the next episode, we'll explore those chemical reactions and see how the similar needs of all organisms manifest themselves as differences in metabolism.
Annenberg Media.
By the National Science Foundation, America's investment in the future. By the American Society for Microbiology. And by the Office of Biological and Environmental Research of the United States Department of Energy. Additional funding was provided by the Arthur Vining Davis Foundations. This program has been made possible in part by the Corporation for Public Broadcasting.
For information about this and other Annenberg Media programs, call 1-800-LEARNER and visit us at www.learner.org.